Hello boys and girls, this is Mr. So again. Welcome back for another lesson of biology. Today the topic is Chapter 26, Basic Genetics on how the DNA do their replication. Before you start watching the video, please remember to like and subscribe my channel. And also, please share the video to your classmate and remind them to do their revision at home. Let's start. Previously on the last lesson, we learned that DNA, there are actually two strands. And it is actually joined by some nitrogenous base in the middle to form a double helix like this. Now the question is, how does this very special double helix DNA makes it a very good genetic materials to hold your genetic information? In order to answer that, I think you can use the idea of a USB. What criteria would you use when you try to buy a USB? When considering the property of a DNA, I think you can think about it as buying a USB. What criteria would you use in buying a USB? Of course, it would be the amount of data that can stored. Right now, I'm using 128 gig USB. Right now, I want to have a very big storage. That is one of the things I consider. So as the DNA, it can store a lot of information. Secondly, the storage should be very stable. If you want to store something in your USB or your DNA, it should last for a very long time without changing. That means stable storage. Last but not least, your DNA, it should also act like a USB with very accurate copy of data. You don't want your data to lose during copying or change during copying, so as the DNA. Now, this is the three important criteria for DNA structure. As what I have said before, DNA is a very large molecules with a double helix, and it has a large number of nucleotides. If you could remember what is nucleotides, this is how a nucleotide looks like, the building block. And with a lot of nucleotides, you can store a lot of genetic information. Your DNA, it should also be a very stable molecules. It should not change. It should store the information very safely. And the double helix structure of the DNA is maintained by many hydrogen bonds. With many hydrogen bonds, they hold the structure very safely and securely. This makes the DNA very stable. And last but not least, you want your DNA to copy and make another DNA very accurately. This requires the help of specific base pairing. As you have learned before, for the base pair, that means the nitrogenous base, A always pair up with T, while C always pair up with G. This is what we call the complementary base pairing. Complementary base pairing. And this base pairing is very accurate. These allow very accurate replication of DNA. Therefore, the DNA information, the genetic information, is passed to the next generation without any changes. And in the following slides, I'm going to show you how exactly does DNA copy themselves to make another DNA. So showing the diagram here, you see a DNA molecules. It has left and right two DNA strands, and in the middle it is held by some very weak hydrogen bond. When DNA replication starts, this hydrogen bond in the very middle of the DNA, it will break. Hydrogen bonds, they are broken, and then the two strands, they will go to the left and right, separate, 分開啦. After the separation, the DNA strand, one on the left, one on the right, they will both act as the template strand. And with the help of a DNA polymerase, this is an enzyme for joining nucleotides. And these DNA polymerase will help to join some new nucleotides into these template strands. So what you're seeing is that the template strand on the left And also the template strand on the right, it allows some new free nucleotide to stick on it. So these are the free nucleotides. They stick on it one by one. And their sticking is according to the complementary base pair, such as A matches with T, A matches with T, G matches with C, G matches with C, etc. And after they are linked together, A and T, A and T, G and C, G and C, the enzyme that I just mentioned, what we call in here, this is a DNA polymerase. It is going to join up the three nucleotides one by one. So right now it's joining up the G and T, and then it's joining up the T and A, and when there are more and more, it will join up furthermore free nucleotides. And this DNA polymerase, it will also join up in the other direction. So for the right hand side, it will join up T and A and C, C and G, G and G, G and T, T and A, joining. With the complementary base pairing, this copying of DNA is very, very accurate. By the action of DNA polymerase, now two new strands will be formed. And now, how many DNA do you have? You have right now from 1 to 2. And now let's take a look at the two DNA strands. The two DNA strands, they are exactly identical, 相同嘅. So let's take a look. TA, TA, AT, AT, CG, CG. So exactly identical. So this is the process of DNA replication. From one DNA making exactly the same copy of DNA from 1 to 2. That's it for the DNA replication part. I hope you enjoyed the video. Please subscribe and like and share the video to your classmates as well. See you next time. Bye bye.